Welcome students to this module on carbohydrates, which is the first unit of your semester 2 chemistry paper, the title of which is Bioorganic and Medicinal Chemistry. We start with carbohydrates as these are the primary biomolecules. Since school, we see that this is the major source of energy giver to the body. For example, for all strenuous work, we need energy, and this energy is provided to us by carbohydrates. If we have a small overview, this is all the food, carbohydrate food, which we find in our daily lives. We have chocolates, gems, sugar, we have wheat bread, we also have oats, barley. Here we have pasta, potatoes, the dairy products. Here we have asparagus or bamboo shoots. We have different types of fruits. It is not important as to the quantity of carbohydrate we consume. It is important to know the type of carbohydrate we eat, which the body needs. So here we have foods which give sucrose. These are all complex carbohydrates. They cannot give us energy as such. So the body processes them, breaks them down into simpler carbohydrates, which the body can use. For example, this is sugar, sucrose, generally called sucrose, breaks down into glucose. This is all fruit sugar, which primarily comprises of fructose. The bamboo shoots, asparagus, have the cellulose. These are dairy products, which primarily have lactose. Potatoes have starch, which give glucose, and so on and so forth. The body consumes the complex carbohydrates, which may be sugar, starch, or fiber. The body, the whole digestive system at various steps breaks it down into simpler sugars, that is the glucose, which is by certain processes stored as glycogen in liver, and it is used by the body on demand. Now, if we see that it generally occurs, these sugars, they are generally occurring in living tissues in form of sugars or starch, basically as glycogen, in form of cellulose in plants, in starch in plants, in tuberous food, sugars in juices, and again in plants. So primarily in plants, we find carbohydrates. And we consume plants, autotrophs, and then in the animal body, it breaks down to release energy. Initially, the classic formula for carbohydrates was CnH2n. But over a period of time, this definition, hydrates of carbon, has changed. For example, we have formaldehyde, which conforms to this formula, but it is not a carbohydrate. Neither is acetic acid, neither is lactic acid. These three compounds, they conform to CnH2n, but they are not carbohydrates. On the other hand, there are certain carbohydrates, like a sugar called rhamnose, which is a carbohydrate, but it does not conform to this formula. So over a period of time, the general formula for carbohydrates, which has now evolved, is CxH2OY, which means that the number of carbon atoms may be different from the number of water molecules. The important factor here is that the ratio of oxygen to hydrogen in carbohydrates is the same as in water. That does not change. Here we have two examples of carbohydrate molecules. Now, this is an aldopentose. You can have an aldotetrose or an aldohexose. Why is this a pentose? Because this molecule has 5 carbon atoms. This is a ketohexose. You can have a ketopentose or a ketotetrose. And why is this a hexose? Because the number of carbon atoms in this molecule is 6. Therefore, hexose. 5, therefore, pentose, penta. The important and new definition is optically active polyhydroxyaldehydes or polyhydroxyketones. So let me first explain why polyhydroxyaldehydes or polyhydroxyketones. If we see a pentose, we see that there are many hydroxy groups here. OH, hydroxy or hydroxyl. So we call it polyhydroxy. And because this aldopentose, as the name suggests, aldo is from aldehyde, this has an aldehyde group. So we call it an aldopentose. It may be a polyhydroxyaldehyde. Here you have 5 OH groups. So it is polyhydroxy again. But since you have a C double bond O, a ketone group, which is attached to 2 groups, it is not an aldehyde group. It is not at the terminal end. It is at the second carbon atom. Aldehyde will always be at the terminal carbon atom. This will never be at the terminal carbon atom. So this is a ketone. And since it is a ketone, we can call it a polyhydroxy ketone. But now the point comes optically active. What is optically active? Optically active is a property of certain molecules when they behave differently towards plain polarized light. When a light of some wavelength falls on them, they will change the direction of that light. They will either, instead of making it go straight, the light will not pass through them straight. It will either turn right or it will turn left. So these molecules are called optically active molecules. This is also a part of stereoisomerism or optical isomerism. So carbohydrates are stereoisomers, optically active compounds, which are polyhydroxyaldehydes or ketones, and they are all those compounds which produce certain compounds on hydrolysis. Like I said, it is important to know the type of carbohydrate we are consuming. So carbohydrates are broadly classified as monosaccharides, oligosaccharides, and polysaccharides. Saccharides is the second name for carbohydrates. We also call them saccharides. Mono means one. When we are talking of one molecule of a carbohydrate, we call it a monosaccharide. It may be 3 carbon atom, triose, 4 carbon atom, tetrose, 5 carbon atom, pentose, or a 6 carbon atom, hexose, or a 7 carbon atom, heptose. But all these may be single molecule of carbohydrate. Then we have oligosaccharides. Now, if we say a disaccharide, it does not mean carbon atoms. It means number of monosaccharides. So if I say a disaccharide, it means sucrose or maltose are sugars which are made up of 2 monosaccharide units. Raffinose is a sugar which is made up of 3 monosaccharide units. Here, I will also like to tell you one other thing. 2 monosaccharide units, when they combine together, will condense together. And they will give out 1 molecule of water. If 3 molecules combine, they will give out 2 molecules of water. So, if X molecules combine, they will give out X minus 1 molecule of water. So, if we have monosaccharide units till 10, then they are called oligosaccharides. The third category is the polysaccharide. So, a polysaccharide unit, there are 2 types. They may be structural or they may be storage. The purpose is different. The structural polysaccharide is a cellulose, and starch and inulin are storage polysaccharides. So if we have monosaccharide units more than 10, then it becomes a polysaccharide. They are high molecular weight carbohydrates. Polysaccharides may further be classified into 2 types. They may be homopolysaccharides or heteropolysaccharides. Homopolysaccharides, as the word suggests, homo is made up of the same type of monosaccharides. They may all be alpha glucose or they may all be beta glucose. And heteropolysaccharides may have different types of monosaccharide units. Examples of homopolysaccharide will be starch and cellulose, and example of heteropolysaccharides will be immunol. Now we bring carbohydrates from a general tone to what we actually should know about. First of all, carbohydrates were identified by Molish in 1930. When he took a compound and added this reagent, 10% alpha-naphthol in ethanol, to that compound. When he added a few drops of sulfuric acid, he found a red-violet ring which was formed at the junction between sulfuric acid and the compound and alpha-naphthol solution. When this is performed actually in class, this is what we get. This is the real test which we get. It is very important for us to understand this identification of carbohydrate. Since we study different class of compounds, this itself is a test which proves that no other class of compound gives this Molish test. If a compound is a starch or a cellulose or a fructose or a glucose or any other carbohydrate, it will give a positive Molish test. Hans Molish was a Czech Australian botanist and he identified this test. So if we prepare a 10% alpha-naphthol solution in ethanol, it is called a Molish reagent. What is 10% solution of ethanol? That is 10% means if we take 100 ml of ethanol, that is 100 ml of ethanol, we add 10 grams of alpha-naphthol to it. We will get a 10% solution. The formation of a red-violet ring confirms that the given compound is a carbohydrate. Now let us come to the structures and also the optical activity. Now here before you is a molecule of glyceraldehyde. Now if you see this carbon atom, this carbon has 4 different functional groups on its 4 valencies: aldehyde, alcohol, methylene alcohol, and hydrogen. This is called a diglyceraldehyde, and this is the mirror image of diglyceraldehyde. If I keep a mirror, then it will be looking like this. Now, the carbon atom in both the cases is asymmetric. We call it asymmetric because the 4 valencies are different. If the 4 valencies are not different, then the carbon is not called asymmetric. Then the carbon is called symmetric. And the other name for symmetry is also chiral. Right? It is also chiral. This is a molecule of dihydroxyacetyl. If you see, this is CH2OH, C double bond O, CH2OH. This carbon is symmetric because the 2 valencies are similar. Why are these 2 molecules being shown to you? If you see this, this has 3 carbon atoms, 1, 2, 3, and this has a chiral carbon. So this is optically active. This is also containing 3 carbon atoms, but this is not optically active. This has an aldehyde group. This has a ketone. Now bringing this here. Now this carbon atom, if I have opened up glyceraldehyde like this carbon atom will not be asymmetric because this carbon has two hydrogen atoms. This carbon atom will also be not asymmetric because two valencies are with oxygen. So only one carbon atom is asymmetric. Here, this carbon atom is not chiral because two oxygen atoms are attached to it, and these two valencies are also similar. Now, this is the parent molecule for all aldose sugars. All those sugars which contain aldehyde, this is the parent molecule. But this is not the parent molecule for ketose sugars because this is symmetric molecule. Now, first thing I have to tell you is that we can grow the carbon chain. The slide is not very clear, but you will still understand that these are three carbon atoms, 1, 2, and 3. Now, if we have to grow the carbon chain, we have to make it 4. Then we have to ensure that carbon 1 is an aldehyde. So the chain will grow from the second carbon atom. You can see that. See, CH2OH and then this red line. CH2OH and then this red line. This will remain the same. So the new carbon atom is coming between the red and the blue. So C1 is an aldehyde and the carbon chain has grown from 3 to 4. This was triose. Now this is tetrose. Aldotriose. This is aldotetrose. Now we have two types here. We have the carbon with highest numeric is symmetrical. That is 4. Here 3. Here 4. Now here you see that in this molecule, OH is on the right side and in triose, OH is on the left side. Why this can happen? Because this is now called conformation isomerism. This is a single bond. This can freely rotate around a single bond like a wheel. It can go right, then it can go left. But if it is on the right and it gets fixed on the right, it will become erythrose. If it is on the left and it becomes fixed on the left, it will be a triose. Now, carbon atoms in between the one aldehyde and the highest are all asymmetric. So carbon atom 1 is not chiral. Carbon atom 4 is not chiral. Carbon atoms 2 and 3 are chiral. Here only carbon atom 2 is chiral. Now chain will grow from 4 to 5. You can see that 4 and 5 are similar. And if we take the case of erythrose, you see OH is on the right. Here OH is on the right. And if we take the case of erythrose, here OH is on the left and here again OH is on the left. Now just observe carefully. Let us take this case. This part of the molecule is similar. If you can see, 3, 4 and 5, 3, 4 and 5 are similar as 2, 3 and 4 of erythrose. The carbon number 2 has changed. In carbon number 2, OH is on the right, and in carbon number 2 in arabinose, OH is on the left. Again, the same logic that it is a single bond, free rotation around a single bond, and if the OH gets fixed to the right, in case of a 5 pentose, it will be a ribose, derived from D-glyceraldehyde erythrose. And if OH is on the left, it will be arabinose, derived from D-glyceraldehyde erythrose. If it is D-glyceraldehyde triose and OH is on the right, it will be xylose. If it is derived from D-glyceraldehyde D-triose and OH is on the left, it will be a lixose. This is the way how we continue to grow the carbon chain? How we identify the number of asymmetric carbon atoms? If we understand these properties, how to draw the structure, and understand that each carbohydrate has an aldehyde group, it will be very important for us to always remember in mind that what are the tests which an aldehyde molecule undergoes. So there are certain tests which an aldehyde molecule undergoes. There are certain tests which a ketone undergoes. But majority of the tests which these molecules undergo are nucleophilic addition type of reactions. In this module, I'm going to deal with primarily 4 types of chemical properties: reduction, oxidation, reducing nature, and action of hydrogen cyanide. Let me first deal with the reduction. The reduction or addition of hydrogen atoms. Reduction may be considered as addition of hydrogen atoms to the carbonyl group. The aldehyde or a ketone will change into an alcohol because hydrogen will be added. For example, in presence of sodium amalgam, aldehyde will change into alcohol. D-glucose will change into sorbitol. Remaining molecule remains exactly the same. From 1, 2, 3, 4, 5, 6, you get CH2OH because 2 hydrogen atoms have been added to aldehyde. If it is a fructose, we get a mixture of sorbitol and mannitol. How interesting. Because here when 2 hydrogen atoms are added, it may be on the right and OH may be added on the left. Just like I said, it is a single bond and there is a free rotation around the single bond. Reduction may also include, if it is addition of hydrogen, it may also be a removal of oxygen. So the carbonyl group changes into, it removes, oxygen is completely removed by hydrogen. So here, the oxygen is completely removed from all the carbon atoms. Aldehyde changes into methyl group. All the CHOH groups change into methylene groups. And even the CH2OH terminal group changes into the methyl group. This reaction happens in presence of red phosphorus and hydrogen iodide. Let me take you back to the previous. Addition of hydrogen will only take place in presence of sodium amalgam. But if we change the catalyst, reduction will take place with the removal of oxygen. So here we have N-hexane and we also get iodohexane. This reaction is very important as it certifies that carbohydrate has a straight chain structure. Always remember this reaction when we discuss the structure of glucose. This is very important for you to remember that this reaction itself shows that carbohydrate is a straight chain molecule. Then we have oxidation. Oxidation naturally means addition of oxygen atoms or removal of hydrogen. So if we treat glucose with bromine water, it is mild oxidation. We get gluconic acid. Only the aldehyde group is oxidized. But if we treat with a strong oxidizing agent like nitric acid, we see that both the terminal carbon atoms are oxidized to carboxylic groups. This is called a saccharic acid. This is also 6 carbon atom. This is also 6 carbon atom. And here, again, we remember that when we say oxidation, when this oxidation occurs in the body, oxidation of glucose, then energy is released in form of ATP, which is adenosine triphosphate. And oxidation in presence of oxygen. Here we are not having oxygen. Here it is nitric acid or it is bromine water. But when glucose is oxidized by pure oxygen, it is called aerobic respiration. And the equation is given by glucose plus oxygen will give carbon dioxide, water, and energy. Then we are not going to get any acid. Acid will only be obtained in presence of bromine water and nitric acid. This is a very interesting and important part of carbohydrate. That is, there are certain carbohydrates, majority of the monosaccharides are reducing in nature, and some of the disaccharides are reducing in nature. Some disaccharides are reducing in nature and some are not reducing in nature. Polysaccharides are not reducing in nature. But what does reducing nature mean? Reducing nature means that certain carbohydrates oxidize themselves. They take oxygen from other compounds and they oxidize themselves into acids. So they will reduce the other reagent. Here I have written a Fehling's reduction reaction where a glucose reacts with Fehling's reagent and forms gluconic acid. Copper sulfate is reduced to cuprous oxide, Cu2. So cupric sulfate is reduced to cuprous oxide. Now, Fehling solution is made up of two types. This is a very interesting laboratory test, and you also have to perform it in your lab to identify a reducing sugar from a non-reducing sugar. So if I give you a compound glucose and I give you starch or I give you sucrose, there is glucose which will be giving you this test, but starch and sucrose will not give you this test. They will not form cuprous oxide. Fehling solution is made up of two types. It has two types of solutions. We mix both A and B to get a Fehling solution. So Fehling solution A is copper sulfate, cupric sulfate, and Fehling solution B is potassium sodium tartrate, solution of sodium potassium tartrate. And when we mix both of them in equal proportion, we get this Fehling solution. Now this Fehling solution, as you can see, will automatically will react to give us this reaction. Now in laboratories, we also perform a test for reducing and non-reducing sugar. Now we have Fehling solution, we have Benedict's solution, and we have Tollens reagent by which we can test reducing sugar. But the simplest is we use it using Fehling's or Benedict's. So reducing sugar gives positive test with reagents. This is the positive test. The colorless solution, when we add copper sulfate and heat, it becomes brick red in color. But if it is a non-reducing sugar, it will not give the test of reducing sugar, and it will remain the color of copper sulfate, which is blue. So by this test, we can identify that the sugar given to us, whether it is reducing or it is not reducing. So these are the various differences. That reducing sugar acts as a reducing sugar because of the presence of a free aldehyde or a ketone. And a non-reducing sugar is a non-reducing agent because it does not have a free aldehyde or a ketone. The reducing sugar will give a positive test with Fehling's, Benedict's, and Tollens reagent, and this will give a negative test. It will give no test. So if we want these sugars to give a test, we have to first heat them with a dilute mineral acid. When we heat them with a dilute mineral acid, the disaccharide will break up into monosaccharide, and then it will give the test for reducing sugar. Glucose, fructose, lactose, maltose, galactose are reducing sugars. Sucrose and starch are non-reducing sugars. All monosaccharides and some disaccharides are reducing sugars, and all polysaccharides are non-reducing sugars. See, this is the test which we get in the lab. These are three reducing sugars, glucose, mannose, and fructose. These two are aldose sugars, and fructose is a ketose sugar. Now, Felling's test results is this is the solution of copper sulfate, which is not changed. This has a slight change. That means very little amount or trace of you have a reducing sugar. Now, if you get a yellow precipitate, that means it is not purely non-reducing. It has some proportion of reducing sugar. And this is the brick red precipitate, which shows that, yes, the given sugar to us is reducing. Now, this is the last reaction which I am dealing in this module, which is the reaction of glucose with hydrogen cyanide. This is a very important reaction which we generally study when we study aldehydes and ketones. So, glucose and HCl react to give glucose cyanohydride. As you can see, the mechanism is very simple. The first shift is when this breaks down HCl, it will break down to give us a cyanide nucleophile and a hydrogen electrophile. With this breakdown, immediately the pi electrons of the carbonyl group shift towards the electronegative oxygen atom, as you can see from the arrow. Now, if the electrons go towards oxygen, what will be the charge on oxygen? It will become negative. The bond will become polar and carbon will become positive. So, oxygen will become the site of electrophile, carbon will become the site of a nucleophile. Now, what is the nucleophile? Cn negative. So, this Cn negative attaches itself to the carbon, as you can see here. Now, oxygen is negative. It is the site for a electrophile. So, the hydrogen atom, it will give its electrons to hydrogen atom. You have to see the direction of atom, arrow. It is very important. Hydrogen does not go to oxygen. The oxygen electrons come to hydrogen. The base of the arrow always shows the electrons, the origin, and this shows the end. And this is how we form the hydrogen cyanide. Now, if I try to show you the reaction mechanism, let me share the screen and I will now see. H, single bond C, triple bond N. This exists as, now you have a lone pair of electrons here, and these electrons come here, making this delta negative and this carbon delta positive. So here you have a negative side and here you have a positive side. As a result, these electrons come to carbon atom and we get H plus and we get a Cn negative. This is the first reaction which takes place. Now, this is the nucleophile. What happens next? In case of a carbonyl group, if I take this is an electronegative atom. As soon as an electrophile comes here, since there is no conjugation here, like in this case, the pi electrons will go to oxygen. This will become delta negative and carbon will become delta positive. Now, this becomes the site for a nucleophile. So, these electrons will be attracted by carbon and we get Cn and here you get O negative, and this is the molecule. This is remaining, this H plus is here. So these electrons are attracted by the proton, and you get H, C, Cn, and here you have OH, and what you get is a cyanohydride. This is a type of an electronucleophilic addition reaction where no new molecule is being produced, and what we get is an addition product. That is why this is called a nucleophile. So this, I hope, covers your 4 properties, and I hope you have understood. Go through this video and please be ready for the feedback. Thank you, students.